Ta-da! There you go. All right, so red blood cell basics. Um, we're going to talk about red blood cells. You may yawn through it, but hopefully I'll get you guys some new information. Everybody's in a different place. We kind of talked about this before that, depending on what background you've come from, you may or may not know even what a red blood cell looks like. I like to think of them as little rafts or donuts. They've got that concave shape. Um, they're round. They have the little dip in the center. Dr. Walsh says they're a donut without a hole, but I think they're great. I was always fascinated by the little flat discs when I was in biology class. I'm like, these are cool looking cells. So everybody knows the job of a red blood cell, right? What's it do? Feel free to chime in.
Carries
Carries
oxygen and
via
iron.
hemoglobin.
Yep. It carries oxygen. Yep. And then after it delivers oxygen, what does it do? Anybody knows the other job? So after it dumps the oxygen, it carries away the CO2. So it doesn't do anything haphazardly. It has a reason for everything it does. And you can't live without oxygen. So the life-giving properties of the red blood cell are really super important. So do you know where they're made?
Spleen.
They're broken down in the spleen once they've served their purpose, but do we know where they get their beginning?
Bone marrow.
In the bone marrow. And do people know how they're made, how they're formed? Like all the We're going to go through it today so you guys will have a very good understanding from start to finish of the whole process. So a red blood cell is a type of cell formed that carries oxygen throughout the body, and it also removes carbon dioxide from your body and transports it back to the lungs for you to exhale. Red blood cells by the numbers. So one red blood cell contains 1 billion molecules of oxygen. They circulate for 120 days, so the average lifespan of a red blood cell. It takes red blood cells 20 seconds to circulate through the body one time. There are 2 to 3 million red blood cells made each second, and there are about 1 million RBCs in one drop of blood. Are those numbers kind of crazy to you? Because they were to me. I was like, wow. Like things are tiny and that there's a lot of stuff, but then when you actually start thinking about it, it kind of blows your mind.
The only thing that I knew was that the red blood cells circulate for 120 days, which is why we only check hemoglobin A1C once a quarter.
Yep. So hemoglobin is heme and globin. So it contains the oxygen. So hemoglobin is made up of those two parts. And then the heme is actually the protoporphyrin protein and iron. And then the iron, if you look over here, so you have the different globin chains. There's an alpha and a beta, and then the iron holds on to the oxygen. So that's we like to think of it as a little car, with 4 seats, with little irons sitting in the seat, and each iron's carrying an oxygen in its lap. If that helps you. So vitamin B6 plays a potentially important role in the incorporation of iron in the protoporphyrin The final step in heme synthesis. Hence, vitamin B6 is an important nutrient for erythropoiesis. Erythropoiesis is the big fancy name for making a red blood cell. Zinc is also a required cofactor for an enzyme that synthesizes the heme. I think it's important when you start thinking about, okay, our body's making these red blood cells, and what are all the pieces that go into it, and so is it a cofactor, or is it actually part of the protein, or where in the process of making this red blood cell do these different pieces come together? But zinc is a cofactor for the enzyme that makes the heme portion of hemoglobin. And copper is a cofactor of enzymes. I had so many typos in this. Sorry, guys. Copper is a cofactor of enzymes essential for iron absorption, transport, and hemoglobin synthesis. So hemoglobin is the protein molecule in red blood cells that carries the oxygen from the lungs to the body's tissue and returns the carbon dioxide from the tissues back into the lungs. The four protein molecules, the globulin chains that are connected together, and a normal adult hemoglobin, abbreviated either HGB or HB molecule, contains the two alpha and the two beta chains. Each chain consists of more than 140 amino acids. And amino acids are the small proteins are broken down into amino acids. Um, so there's 140 amino acids in each of those protein chains. Each globulin chain contains an important iron-containing porphyrin compound termed heme. Embedded within the heme compound is an iron atom that is vital in transporting oxygen and carbon dioxide in the blood. The iron contained in hemoglobin is also responsible for the red color of the blood. Each human red blood cell contains 270 million of these hemoglobin molecules. So 270 million of these little 4-chain hemoglobin molecules. It's kind of crazy. This is just another way to look at it. You've got 4 subunits. The protein is the globulin. And then you've got the non-protein heme, which is the iron, and the protoporphyrin. And then you've got alpha and beta. So red blood cell production starts in the bone marrow from a hematopoietic stem cell and then goes through nuclear maturation. So you're looking at about 3 days for it to go from a pro-erythroblast to a basophil erythroblast to this polychromatic. And as you see, it's getting redder and redder and redder, right? And then the nucleus gets smaller and smaller and smaller until it's ejected at the edge of the bone to a reticulocyte, and then this little nucleus, and then into a fully mature erythrocyte. So when we're looking at, we're going to go into when you're looking at the different lab markers, what are they and what do they mean? And, is the cell big, right? Or is it small? Like what's going on in this process that is making a good red blood cell? All right. So all blood cells are made in the bone marrow. This is the red spongy part inside the bones. Red blood cells arise from a single type of cell called a hematopoietic stem cell. There are 3 basic types of blood cells: white blood cells, which fight infection, platelets that stop bleeding, and red blood cells, which carry oxygen and carbon dioxide through the body in the form of hemoglobin. If the hemoglobin is low for any reason, then the result is anemia. So when we get a blood sample, and we talked about this with dehydration, right? When we get a blood sample, and they put it in the tube and then they centrifuge it out, you get a portion that's red blood cells. These are the erythrocytes. It's going to be between 38 and 48% of a whole blood sample. These cells keep tissue alive by bringing oxygen, and then you're going to have platelets. The thrombocytes constitute less than 1% of whole blood. These form the clots. And then you're going to have a small portion that are white blood cells. They're less than 1% of the sample. And then you're going to have the plasma, which is the fluid. which is 52 to 60% of whole blood. So when we talk about a dehydrated blood sample, it's a sample that doesn't have as much of the plasma. So it, when they always draw and take a very specific amount of blood. And then when they measure that amount of blood, they're going to, if there's not as much plasma, then this is going to be elevated. The amount of red blood cells. It's only a factor of concentration. Any questions so far? We good? I don't want to go too fast. So get some of my screens out of the way here. All right.
No, Jill, I find it so interesting the nucleus is kicked out because I always think of a cell having a nucleus, and that's
Mm-hmm.
fascinating to me.
Yeah, in a red, I mean, I believe it's the only cell in the body that doesn't have, well, other than, I think, sperm and eggs have half, right? Half the gametes and things. But I think it's the only one that doesn't have a full nucleus. So that is the thing. All right. So the plasma has the anticoagulants needed for purification. It can be prepared as soon as it's been mixed thoroughly. There's fibrinogen, there's platelets and cells, um, and then the composition of ions is representative of the circulating blood. It's considered less stable for longer storage. So when you get like a plasma sample versus like a serum sample, in a serum, the anticoagulants aren't needed. They delay it for 30 minutes, so the clot forms. Fibrinogen is absent. It's a cleaner sample. It's depleted of cells and cell remnants. Um, but the latent clotting can lead to fibrin formation. Clot retraction elevates potassium levels relative to the plasma value, and it's considered more stable and the gold standard for biobanking. Um, so there are the different, when you're getting samples, just so you guys know what the difference is on those two. Erythropoietin is a hormone that is produced predominantly by specialized cells in the kidney. So once it's made, it acts on the red blood cells to protect them against destruction. And at the same time, it stimulates the cells of the bone marrow to increase the production of red blood cells. So like everything else in our body, red blood cells are controlled by hormones. Um, Erythropoiesis requires certain minerals and metals, iron, copper, zinc, and manganese, and it requires the vitamins B12, folate, C, E, B6, thiamine, and which is B1 and B2. It requires proteins, which makes the globulins, which are amino acids from the liver and the hormone produced by the kidneys. So erythropoietin, which stimulates the red blood cell production from the bone marrow. All these big fancy words. And I'm not going to make you spell them. Just look it up. Every time I typed this in the slide, it told me it was wrong. And I had to keep going back. Very annoying. Canva does not know biochemistry. So to make heme, you're going to need B6, zinc, and iron. And if you can't make heme, you can't make hemoglobin. So if the hemoglobin is low for any reason, the result is anemia, right? So specialized cells in the kidney are capable of detecting and responding to low levels of oxygen through increased production of erythropoietin. So when there's sufficient oxygen in the blood circulation, the production of erythropoietin is reduced. But when oxygen levels go down, the production of erythropoietin goes up. This is adaptive because it facilitates the production of more red blood cells to transport more oxygen around the body, thus raising oxygen levels in the tissues. I always think the body is so amazing and fascinating. So if you have too little erythropoietin, which is usually caused by a chronic kidney disease, there will be fewer red blood cells and you will have anemia. And anemia is a symptom. I don't know how many times, you know, we treat things as a disease, especially in modern medicine, right? But anemia is just a symptom. It's not a disease, and it's not enough information to tell us the underlying cause. So therefore, we can't just say anemia and understand why it's present from just that one word, right? We've learned about macrocytic, microcytic, all those things. So, once again, if you're looking at this, at what point in this process, do the different types of anemia come into play?
Well, Joe, I just want to say I'm appreciating that kidney comment, like that kidney disease could result in anemia because I never would have thought of that. And I mean, I guess it would be easy to assume that in more of a aged population, but for anybody that had any kind of chronic or genetic kidney disease, or
Mm-hmm.
I have a client that I see for physical therapy who has had a kidney removed. And while I wouldn't, there's a reason behind that, but even just having one kidney and just her body's ability to use those negative and positive feedback loops to, manage these other components of red blood cells. It's fascinating.
Yeah, you start to, this Jen always said, right? If you don't understand the process, right, how are you going to jump in here and really take a look at the entire picture and all of the connections? If you don't really understand how a red blood cell is made and you're just taught to look at the Weatherby book, right? And say, oh, low red blood cells means XYZ. Well, why? High red blood cells means this. What's the other, like why? Like what is the MCV? What is it? We're going to get into that. I mean, they're calculations. Like there's a way to figure these things out. Um, so I think it's really important, that you understand, what are these steps that, go from here to here and what's needed, right? Because I mean, back here on this other slide, we talked about way back here, what's required. Um, sorry, getting dizzy, like flipping back through. Vitamin B6. It plays a role in incorporating the iron into the protoporphyrin and the final step in heme synthesis. So if you don't have B6, zinc is needed to make the heme portion. Copper is essential for hemoglobin synthesis. So if we scroll all the way back up, right, um, and we're looking at this again, in order to go from, ejecting this cell and getting that heme, right? I mean, the heme's being formed all the way along here. Every time it's getting redder, right, that's hemoglobin. And if you don't have enough to make those 270 bits of hemoglobin that go into this cell, and maybe you've only made 100, right? That cell's not going to be as red. You could have all the iron in the world, but if you don't have B6, zinc, and copper, you're not going to end up with a nice, pretty hemoglobin-filled cell. So, these are the markers we're looking at. We're looking at red blood cell numbers. We're looking at hemoglobin, hematocrit, MCV, MCH, MCHC, and RDW. And how many of you could tell me exactly what all those letters stand for? Anybody not sure? It's okay. Without looking it up, have you ever like gone through and figured out what each of these stand for? You don't have to embarrass yourself. So the RBC number is how many red blood cells do you have? So when you get that sample and you get that, separation, how many red blood cells are in a volume of the volume of the sample? The hemoglobin is a molecule. It's measured by weight and measures how much hemoglobin your red blood cells contain. Remember, each blood cell should have 270 million of these little ones. So that number is measuring how many are in the sample. Low
Okay,
RBC.
okay, hold on, sorry.
Okay.
So
Okay.
go back to that one. So if someone's hemoglobin is low,
Yeah.
that's going to tell us they have whatever, 170 million of these instead of 270, right? I mean, if 270 is
Yeah.
the
I mean,
optimal. Yeah.
there's
Yeah.
no like, if it's 11 point whatever, that means, yeah.
Yeah, but I mean, it's just, I don't know. This is just giving me a visual that I, did not think about in the least before of like, okay, I know hemoglobin low, bad, whatever, want it better. But what does that mean actually in that cellular level is,
Right.
yeah,
Well,
that thing is kind of empty.
for every hemoglobin they're missing, that's 4 oxygen molecules they can't carry,
Yeah.
4 carbon dioxide molecules they can't get rid of, right?
Yeah. Wow. It's a real detriment.
Yeah. So low RBCs, what are some reasons why you might see low RBCs? Some of you probably read the slide that I'm about to click to, but what do you think if you see low? We always look at high, right? RBCs are high, we're dehydrated, move on. But what happens when you see
Toxicity.
toxicity?
Yeah.
Okay, what else might cause a low RBC? Put on your thinking caps.
You don't have the nutrients to make it.
Okay.
Hemorrhaging and destruction of the RBC for some reason.
Mm-hmm. So nutrient deficiencies, toxicity. What about in women? Why would you see a low RBC in a woman?
Higher blood
Higher blood.
flows, like menstrual cycles.
Yeah. Yeah. Someone has really heavy periods. They really bleed a lot during their cycle, depending on when, you get that count. And if they're doing it month over month over month over month, you can see chronically low RBCs. A lot of times ulcers or like mini gastric bleeds or things like that, you'll see low RBCs. Let's see what we got and what we missed. So blood loss, heavy menstruation, bleeding from a stomach ulcer, definitely nutrient deficiencies, low iron levels, RBCs being destroyed before they're replaced, the body just making few RBCs because of those deficiencies. And then again, once again, having chronic kidney disease, right? Would all cause the body to have low RBCs.
Joe,
What?
so like I'm following everything that you're saying, but I
Mhm.
obviously can't remember everything that you're saying.
Yep.
So these slides, like, are they going to be housed in Kartra or like where we can download them and they'll be searchable, or will we be able to search them somehow?
I can give you copies of the slides.
Okay, so they'll be in Kartra or they'll be in Telegram, like,
I
or
will
we don't know
probably
yet.
put them in a Google Drive for you.
Okay, because I think it would be good to be able to download them like into my Notability. So, like,
Right.
okay, I have a problem with red blood cells. Now let me think of everything that I need to think about now when I'm focused on it. Does that make sense what I'm saying?
Yep. Yep. I have no problem with that. All right. Hematocrit is the percent of packed cells in centrifuged blood. And if you're not making red blood cells, the hematocrit could be low outside the functional range. MCV is mean corpuscular volume. So a corpuscle is just a cell, right? So it's the mean volume in a single red blood cell. MCH is mean corpuscular hemoglobin. MCHC stands for mean corpuscular hemoglobin concentration. And these are just figured from the RBC and the hematocrit. And the hemoglobin numbers. So MCV is equal to the volume of a single red blood cell or its size, right? It's the hematocrit times 10 divided by the RBC number. MCH is how much hemoglobin is in a single red blood cell, or how the weight of it. So it's the hemoglobin times 100 divided by the RBC number. And the MCHC is the calculation of the concentration of hemoglobin in a red blood cell. So it's the hemoglobin times 100 divided by the hematocrit. So how much, or you can think of it this way, how much hemoglobin is in the red blood cell relative to its size? And these are the numbers that can help you determine what kind of anemia is present. Any questions on the slide? I was shocked to find that these were just calculations, like what the actual math was going into each one of these. So RDW is typically high RDW is typically nutrient based. Over the course of 120 days, new cells are constantly being produced. Therefore, the variance in size will increase. This is the red blood cell distribution. I think it's with RDW. So it's telling you how many of the cells are small, and it's just an average. Small, are they all small? They all, are there lots of different sizes? That they all, are you putting out perfect red blood cells consistently, or are there small ones and big ones? Is there a big variation? It's a mark of the variation. So low or normal means there's not much variance in size. Low or normal can also mean it's a severe deficiency. So if you're so deficient in a single nutrient that you're constantly putting out large cells, and this has happened over the entire course of the last 120 days or whatever, then your RDW is going to be normal. There's not going to be a lot of variation in that size. Ooh, I went backwards. Sorry, guys. So low production, destruction, or loss can then equal anemia. Destruction is not going to change the variance in size, so the RDW could be normal. And loss is going to lower the number of RBCs, but it's not going to have a bearing on the size. So you could also have a normal RDW.
Can we talk for a second about what destruction means or what that looks like? What's happening? I mean, spleen breaks the red blood cell down, right? But is there another way they're destructed?
If you don't have good cell membrane integrity, they could not last, right? If you're not making good cellular membranes for whatever reason, the cells come out. Inflammation could damage them. Toxicity can damage them, right? What are the causes of other cell destruction in other parts of the body?
So much to think about.
I know. There is. So RBC, hemoglobin, and hematocrit tell you that anemia tendency is there, right? So any of these numbers, it's measuring an amount. So when you look at RBC numbers or hemoglobin numbers and hematocrit numbers, they're measuring amounts. So having a low hemoglobin, right, can tell you that there's anemia. But the MCV, MCH, and MCHC can tell you why the anemia is there because of the size and the color. And there's a whole nother class that we break down, okay, what does this mean? Like when you're looking at numbers and you're looking at samples, how do you really look at the numbers and figure it out. So as you go down, right, you can have large cells that have a lot of color, right? Like if this is normal, right? If this is normal coloring, and then like, so this one in the middle is a perfect red blood cell, right? So you can have them that are too red, too much, um, too big or too small, and then you can have ones that are pale and too big and too small. So you can have all these different sizes in a blood sample based on, you know, whether or not something was available at the time the red blood cell was made. Did you guys ever think, like actually visualize like all the different ways red blood cells can come off the line if things were missing?
I don't think I've ever thought about anything. My brain doesn't work a lot of times. You got to understand, right? My background was basically here is how it is, and this is how it is. Like
Yeah. Well,
there wasn't a lot of encouraging of critical thinking or thinking outside the box, etc.
no, and I mean, and it's helped me so much to realize like I can literally visualize this. Like I can visualize these red blood cells coming off this assembly line, right? And not having iron, not having, the zinc to put the heme in, right? And just having red blood cells come off the line with not the heme and not enough hemoglobin and not enough iron or missing this and not ejecting the nucleus because it doesn't have what it needs to and it being, you know, pale and big and all the
But
things.
but this is also like how you really become a problem solver and
Yes.
getting down to that root instead of just throwing more things just to cover up things.
Yeah. Yeah. There's a whole thing, I think, if on the Wednesday call I said, the don't be a people parrot. I think we're learning, sometimes we learn so many things, and it's like being in kindergarten, first grade, and you're just learning your ABCs, right? And you have no idea what an A is, what it sounds like. You're learning your ABCs, right? You're a toddler and you're going A, B, C, D because you learned the song, right? You have no idea their letters. You don't know that they make sounds. You don't know what happens when you put them in words. Like you have no idea of the language. You just know the pieces. And sometimes, coming up through functional medicine school, it's the same thing. We can say RBC. We can say MCV is high. MCV is high, so they've got to be low in B12, right? And then you get your first set of labs back as a brand new functional person, and they have, a raging high MCV, and they have, their B12 levels are off the charts high. And you're like, I thought they were supposed to have low B12, right? And you're looking at labs going, I don't understand. How's that possible? So, like those are the situations where you need to understand. Like so here we go. For heme to be created, B6 and zinc are required. The polyporphyrin ring also needs to have iron attached to finish creating heme, which can now attach to globin to make hemoglobin. Without these three nutrients, heme cannot be made, and therefore no hemoglobin is made, so the reticulocyte ends up being too small, microcytic, and it lacks in any oxygen binding properties. It's usually very pale and hypochromic. So, we end up with this cell, and instead of it getting small and red and whatever, now we have a cell that doesn't have enough hemoglobin, right? And it's just this little teeny tiny pale thing. So sad looking, isn't it? So if you have low hemoglobin, look for B zinc and iron deficiencies.
I just have to laugh. I feel like it looks like a ghost. And when you have someone that really is really anemic, they're so
Yeah.
pale.
Yeah. It's exactly what's going on. I mean, it's we want to be like so savvy sometimes, and then sometimes it just comes back down to, this. It's kind of like when you saw the first time I saw the different vegetables and how they looked like different parts of the body. And you cut a carrot and it looks like an iris from your eye, and the cauliflower looks like a brain, and the inside of a tomato looks like a heart, and the chambers of a heart, and you just go, and each of those vegetables contain vitamins that support that organ. I'm like, you don't need to be a rocket scientist to know that these things are meant, like, designed to help these organs.
I'm so glad you're homeschooling us, Joe. I didn't know this.
I
You know,
didn't know that either. That's cool.
yeah, it is really cool. And then you go like, yeah, you have pale red blood cells, teeny tiny pale red blood cells. You look like a teeny tiny little pale ghost. You're just a little withered, like, yeah. So low iron is going to make RBCs smaller, and low B12 or folate is going to make RBCs bigger. And how is this going to affect the RDW? Anybody want to take a guess? We kind of touched on it in the slide before, so this isn't a trick question. So let's say somebody has low iron and they just went vegan. It's only been a couple months. I don't know who was trying to talk. You were muffled. I have to call on someone.
Well, you said that the RDW is with, right? So I'm picturing, are you asking the
The
question about,
RDW, the RDW is the number that says there's variation in the red blood cells, the distribution of width. So it's a variation number. So if it's low or normal, that means there's not a lot of variation. And if it's higher, that means there's a lot. So someone's a new vegan or vegetarian. They've sworn off all red meat in the last 2 months, and they get their blood work drawn. And what are we going to see possibly with the RDW?
well, I think that could be high then because they have leftovers of like what would have been maybe normal for them. And then they're going to have these new ones that would theoretically be small because they're not having those cofactors and, you know, from what they're eating.
Right. So now let's take that person down the road 6, 7, 8 months. They go back in, they get another blood draw. We look at their RDW, and what's it look like?
Well, and so now it's not going to be as large because now they've had, and in the 120 days, red blood cells have all turned over, right? So now they have like one baseline instead of, I'll call it two, where they had more food variability. Now they're more consistent in their nutrient intake, which is insufficient for them to be making the right size of the red blood cell. So
Yeah.
it would look better theoretically. The RTW would look better, probably be normal, but it's not really normal because it's just saying it's normal from this 120-day segment, like crap in, crap out, right? I mean,
Right. And I mean, you could, what if it is shockingly average? We know that they just turned vegan. It's been 2 months and their RDW is smack dab in normal range. But you know that they have a deficiency because their iron is low, their B12 is low. What could be happening with that sample? Thinking caps are on.
So I don't want to steal the stage here, so I'm
No,
trying
go
to
ahead.
get people to jump in. But so you're saying it would look normal, but I think then it's still just an average, right? So I mean, at that
Ding, ding,
point,
ding.
it's just a matter of equal amounts of bigger and smaller,
Yep.
balancing out to look
Yeah.
great.
Yeah. So, I mean, this is where we have to look at the whole picture, right? We can't get stuck on one number. Um, this is where you could take, a perfectly normal number and go, this is hiding something, right? Just like when you look at a dehydrated sample and you mistakenly think it's a normal sample, and then you go, oh, this is great, right? Great hemoglobin, but the RBCs are 5.2. So it's going to look like there's more hemoglobin than there is. So,
Hey, Joe, in your experience at this stage of the game, like, and there's not going to be an exact answer, but it's like, how much do you weight the markers? Like let's say you have one or two markers that look um good, but you're suspicious that they're not really good. I mean,
it's
weighing that
true.
out versus the symptoms and that sort of thing. Like how you,
This is where you really have to know your client. I mean, it's one thing to look at a set of numbers, right? It's another thing to look at, you know, who is this person that has this number, right? A normal RDW with someone who's eating a wide variety of stuff, whose other markers are normal, I'm not going to second guess it, right? But if it's like looking at an RBC that's good, but knowing that this person detests drinking water and drinks coffee all day and soda. I know they're not hydrated. I mean, right? You have to know the person. And the more you look at labs and the better you know your client, the more you're going to get a sense, and that's where it's just another piece of the puzzle, right? Like switching a number just to switch a number, like you can't get that attached to the number. Um, you just have to start to get a feel for what is this number telling me about this person. Um, does that make sense? I know it's not a perfect answer, but I mean, you
no,
just
it
start,
really does.
yeah,
Yeah.
you
Thank
just
you.
start getting a sense of, how these things relate. And the more labs you look at and the more you jump in the pool, right? Like Tracy says, and get that practice, the more you'll start to understand. So, um, how looking at a low iron, what's that going to do to the MCV, MCH, and MCHC? Right? These are volume numbers and color numbers, right? So when you look at the MCV, which is a volume number, right? It's how big it is. Low iron is going to make the RBC smaller. So what's going to happen to the MCV? Okay, smaller red blood cells, smaller MCV, right? Go ahead, Kristen. You unmuted. Were you going to say something?
No, you just said it. I didn't hit the button fast enough because I thought that would be smaller then.
Yeah.
Yeah.
So how about the next one? MCH. Low iron is going to make RBC smaller. What's going to happen to the mean corpuscular hemoglobin if we don't have iron?
That would go down too, right? Because you need the iron to make hemoglobin.
Right. And then the MCHC, which is the mean corpuscular hemoglobin concentration. If the cell is smaller, I mean, this may This might go down because it's the concentration number. So if it's smaller and the hemoglobin is smaller, you might have more of a normal MCHC. It may not.
Okay, so question, when you use the word concentration, I know what you're talking about here, but I'm muddying the waters a little bit. If someone's dehydrated and so things might be elevated and concentrated, how does that come into play here?
So this is
Besides
the
you can-
concentration within the red blood cell. So it's how much hemoglobin is in the cell compared to the size of the cell. So it's a comparison of MCV to MCH.
So dehydration
So
wouldn't have anything to do with this then?
not as much as it does like an RBC number. I
Okay,
mean,
thanks.
because well, think about it, right? So if you've got low iron, right, you're going to be making smaller cells because it's not going to have as much hemoglobin in it. So the cell size is going to be smaller because the hemoglobin is less. So if these go down at the same rate, the cell size and the hemoglobin at the same rate, the MCHC is just averaging these two, right? It's more of a ratio kind of number. So the mean corpuscular hemoglobin concentration might not change because you have a smaller cell with less hemoglobin. If you had a bigger cell with less hemoglobin, right, your concentration's not going to be much at all. But if you have a smaller cell with a normal amount of hemoglobin, you're going to have a higher hemoglobin concentration for the size of the cell. So it's the amount of hemoglobin compared to the size of the cell.
Okay, my brain just got tied in a knot. If you if you have say there's a list and one is a normal MCHC, one is a low MCHC, and one is a high MCHC. Tell me where I go with those because I got confused there.
Well, go back to uh sorry. So MCHC is the concentration of hemoglobin in the cell. So it's the hemoglobin divided by the hematocrit. So how much hemoglobin is in the red blood cell relative to its size is what this is answering for you. How much hemoglobin is in the cell compared to the size of the cell?
So if you were looking at the MCHC and pondering it, you'd want to go back then and see where your hemoglobin is and where your hematocrit is.
Mm-hmm.
Okay.
Right. Because
I don't know why I get so confused.
the MCV is going to be the hematocrit times 10 divided by the number of RBCs, right? So that's the volume of the cell. Because the hematocrit is a concentration number 2, right? If we go back to hematocrit is the percentage of packed cells in centrifuge blood. So if you're not making RBCs, the hematocrit could be low. So the hematocrit is going to tell you how many cells are packed in there. So then when you're looking at this and you're dividing the hemoglobin divided by the cells, it's going to tell you how much hemoglobin is in a cell relative to the size of the cell. So then you go, you start thinking about, here's our cells, right? So what's this cell, right? Which is big and pale versus this cell, which is tiny. Oops, sorry. Tiny and really red. So there's a lot of hemoglobin in a tiny cell. What's going to happen to the MCHC? Lots of hemoglobin, little cell. What's the concentration?
be high,
It would
right?
be high. So an MCHC would be high versus this one. You have a really big cell with not a lot of hemoglobin. The MCHC is going to be what?
Lower.
Lower.
That helped. Thank you.
Yep. So low iron is going to give you a low RBC, a low hemoglobin, low hematocrit, right? And, these are going to be low and you're going to have an elevated RDW. And a low B12 and a low folate, you're going to have you could have low RBCs, low hemoglobin, low hematocrit, but you're going to have high MCV, high MCH, and high MCHC. And in all of the cases, you could have an elevated RDW. So let's think. On the production side, if you've got a low RBC, low hemoglobin, and low hematocrit, those are all production issues, right? You're not producing enough. You could have low iron, you could have low B12, you could have low B6, something involved with the actual synthesis of the red blood cells. So in the destruction part, cells are going to last 120 days. They can be broken down too early due to inflammation or the breaking down of the membrane. And then in a loss situation, you're looking at menstrual cycles, microbleeds, gastric ulcer. So those are those. So here we go. Here is a 39-year-old female. Her iron numbers are older, so they weren't run at the same time, but she has a lowish ferritin of 54 and a higher iron saturation of 38. Perfect iron saturation is 30, okay? The MCV looks like it points to a need for B12 and folate, and the cell might be on the large side. But at the same time, her hemoglobin and hematocrit might be pointing to some type of nutrition deficiency as well. More B6, more zinc, more iron, and a need for materials to make heme. But the MCH and the MCHC look okay. Can a need for B12 and folate falsely increase MCH and MCHC when there's actually some sort of anemia happening? Before I even go further, remember what happens when we don't have when we don't, where would we go? Sorry, scrolling all the way back. So what happens when we don't have B12 or folate? What kind of anemia?
That's going to be macrocytic, right?
Right. So you're going to have bigger, right? These cells are going to be bigger. Um, right. So we're going to have bigger cells. Wish I could put these side by side for you. So B6 and zinc could give you microcytic, right? Without hemoglobin being made. Here we go. So when numbers don't match, right? You've got a lower RBC number, right? Hemoglobin, hematocrit, you may have a little bit of deficiency there, but when numbers start not matching up, there could be competing deficiencies going on, and you can have all these different cells in a blood sample, and then averaging things out, and numbers can hide the truth. Um, so it could be a need for several nutrients. Um, can you have big hypochromic cells, right? Which, where's the chart? We can, right? They can be big and hyperchromic, you know, not have enough heme iron, not be carrying enough, but still be big. That can happen. Back and forth. So this might be an example of iron floating around and not having enough bees to make heme. Personally, without, I would have new samples run that were, so you're comparing apples to apples to apples, right? Because you've got old data with new data, and you really, I mean, you really can't understand the numbers if you don't have current data. So, any questions? How would you look at these numbers? Any of you ever gotten labs and just totally scratched your head on them? Like, I don't understand. What is the body doing?
Well, for me right now, I'm sort of thinking like, on quick glances, you look at things and it says normal, and I'm like, yay. And now I'm like, damn.
Right. It's you have to look at I think that all the time. I get people that I know, I look at my dad's blood work and the doctor checks it off and they go, everything's fine, it looks normal. And I put it in my thing. I'm like, you really only have a couple things out of whack, Dad. But then you start looking deeper because I know doesn't eat red meat. Like I know like these things are happening. I know that this can't be right. Um, so you have to look at the whole person. You can't just look at numbers at face value and go, yay,